that side of things. But
did you put together the whale fall one?
no, I did not. I just know that's the program that they use, and this summer I spent some time doing some photogrammetry and stereo camera work.
Cool.
So I just got a little bit of experience with it.
Cool.
Have the program.
Yeah, that's who introduced me to Colmap was K2.
Yeah,
So their lab's been using it a bit.
Chris Roman's lab at URI. They do a lot of it.
What do you call the program again?
Colmap.
COLMAP.
Yeah.
Like cool, but without the extra O?
Yeah, so it's not that cool.
Cool map. That would have been I think, better choice of a name.
Yeah.
Cool map.
That sounds a little...
Write that down.
Yeah.
Sounds cheesy, like not professional.
Colmap is just obscure enough to work.
Yeah. We kind of been low here before, haven't we?
Yeah.
I can skip over this part.
Yeah, I think that.
Or go a little faster here.
Right.
That's that one rock that it's just about to hit. If we get past that one, I can drop back down again.
Mm-hmm.
Yep, that's the one we hit last time.
Been here before.
There's two of them right here, but I think I can hop over them and drop back down.
I wonder if the rockfish moving around are going to cause the photogrammetry software to have fits with the fish in different places.
Yeah, I don't know if you saw the whale fall one, but some of the octopus, I mean, it was covered. Some of them made it into the model and others didn't. I guess just depending on how stationary they were.
Interesting.
Did they have extra legs?
They were mostly just purple blobs. It's not that finely resolved.
It's a cool angle.
Seem to be stuck. Stuck again. Does that boot sponge have a new list?
We'll find out.
Let's see how that shows up in the model.
Yeah.
I kind of barely bumped it with the.
It seemed to rebound.
They're tougher than they look.
I just touched it with a bumper going over once and I backed up and came around it. Okay, I think this is where we started, isn't it?
Yeah.
Yep.
Good.
Yeah, unless I'm good. Okay.
Looks good.
Lizzie says, looks great. Thank you all. I think she's still having problems with her V-Link.
We're gonna Jake's gonna do some file housekeeping here and start the DSC up again.
Okay.
So I'm going to zoom out a bit on HiPAC. We can get some context for the
You're
next
gonna
bit.
keep the mosaic stuff in a separate folder, right?
Yeah, it's
You don't want to add a whole bunch
Very-
to that folder.
that's
Oh, okay. Never mind. I'll stop looking over your shoulder. Sorry.
Okay, so I have up, Steve, do you have HiPAC on channel 3?
I do.
Okay, on HiPAC, I have our location, which is near 82. And then we've got a ridge feature to follow either along the top, or if we choose, we can go along one of the sides to 83. And then this is obviously going to happen over the next watch. That 83 is about 600 meters away, and then from 83 to 84 is another 700. So they have potential to get at least to 84 if, depending on if we're trucking along. Gonna have to discuss whether we're prioritizing getting the ground coverage or stopping for more stuff. I think 85 would be a stretch if we're pulling up at 8. That's another 800 meters away from 84. It's also downslope, so we'd have to drop off, I think.
I don't think we're going to be stopping as much to collect samples from here on, just because we've already filled up on so many sponges. If we see 1 or 2 target species of coral, I think that'll be the only thing, other than also possibly Niskin bottles. And actually, before we take off here, getting a Niskin bottle to correspond to this photo mosaic would be probably a great thing to do. So before we take off, let's take one here.
Sure.
And
so if you were to go 0.2 knots from 82 to 83 to 84 without stopping, going 0.2 knots, it would be 3 hours and 20 minutes.
okay, I don't know that we really need to make it all the way to 85, but yeah, we'll start heading that direction over towards 84 and 83 first, and we can start a transect when we do that.
Sorry, what
Yeah,
did you
Lizzie
say
just
about
said-
preference for 85?
Lizzie just chimed in, there's no need to make it all the way to 85, so we'll just continue along these lines towards 83 and then 84 and just make it as far as we make it.
Okay, sounds good. Yeah, so that time estimate of 3.5 hours is to 84 going 0.2 knots. So if you go 0.3, you make it a little quicker. 0.2 gives you time to stop and look at stuff without.
Meredith Everett, who's one of the leads right now, has been chatting. She's from the Northwest Fisheries Science Center, and she's going to filter the water and she's going to extract the environmental DNA to see what we're seeing on screen. Does that match with what it's sloughing off into the water? Are there other corals and sponges out there that we're not seeing?
So how come you don't get to listen to their audio right
I have one in case they need me, but they were doing a photo mosaic for so long. It just sometimes there's just nothing for me to say. So
Sorry, was that a sample I missed there?
Yeah, that was sample. So,
Yep.
Dan, was that Niskin number 5?
It was, yeah.
Okay. I think
no,
I
I
think
was
number
really
4 was empty, but
Can you tell Lizzie to get her mic?
Yeah.
yeah.
Lizzie, could you mute yourself while you do this
Lizzie.
the switch over?
There we go. Okay, so sorry, what was that sample number there?
083.
Which way are we gonna head out from here?
We'll be going
in
roughly
general.
northwest.
Northwest.
And can we quickly zoom on that sponge, the kind of center right bottom, right as we take off?
I was gonna do some tether management so I can zoom before
sure.
I lift up. What sponge?
Sure, it's now in the bottom right-hand corner.
Zoom in there a bit, Steve. Not great.
We get the lasers back on.
Ooh,
Yeah, we think that's a polymastia sponge.
Jay, can you get the lasers on?
Abby says, perfect, thanks.
Thanks.
a little king hat.
Koopa shell.
She said it's one we'll be looking for on a future dive.
hey y'all, this is Lizzie. I'm back. Sorry about that. Just FYI, for this sponge in this depth range, we are hoping to dive again when we come back to Channel Islands in this depth range at Footprint Reef. So it should be pretty similar habitat.
Okay, great.
So if we can't and don't have enough room to collect it here, hopefully the next dive on Footprint Reef we'll be able to put that on the list. And if you want, Abby, on that dive plan, make a note that you really want that sponge.
We can keep an eye out for it at San Lucia tomorrow too.
Yes. Sorry about that audio earlier. The Deputy Superintendent of Channel Islands National Marine Sanctuary says, hello, everyone. He popped into the conference center that I'm in.
Afternoon, Deputy.
Thanks for having us.
He has been watching and listening and sharing screenshots all day long. He just keeps one dedicated monitor with the sound off all day. So he's really excited about our operations, and
Awesome.
we're excited to have him.
Northwest, you said, right?
Yeah, I actually think we'll be going 315 dead on.
How many Niskins do we have left?
One
One, okay.
Niskin.
On here, I have that we haven't fired number 4 or 6. So I think we just skipped past number 4.
No, we have number 4. We fired it.
You did? Okay,
Yep.
let me check my sample sheets.
It was us that fired it, actually.
It was? Okay.
Yeah.
Looking at
Oh,
the,
yeah,
At the
yeah.
end of Transect 9.
Yes, it was. Thank
looking
you.
at the live video of it. That's
And that was sample number 82.
Yeah.
I did a countdown in my head for that Niskin.
So pretty much for the rest of this dive, Meredith, do you foresee wanting to collect any of the corals that we've been encountering so far?
Only if we come across some of the Plumarella again, but otherwise we can just proceed on transit.
Yeah. Yeah, I think we should focus on transects and then opportunistically looking for another target for a 3D model that might be a little bit smaller than the one that we just did
Sorry.
with some more vertical height.
And we actually only have 20 minutes left in this watch, and then we'll be handing it off to Chad and a few others to finish this out.
Yeah, we'll be in good hands with Chad in terms of selecting other potential targets for photo mosaics.
Yeah, it'll be Chad and Lisa and Jen B.
A little racetrack.
Totally. Should we fly right over the top of this or offset? Seen it from one side, seen it from the other.
Yeah, we can see it from the top view.
Yeah, you happy with that?
Bridge nav.
It's a mini mosaic.
100 meters, 315, speed 0.2. Thank you.
Definitely, I'm going to have to get a geology paper about this area, do some reading.
Definitely. I was hoping that Nancy Prouty might be joining us for some of these watches, who might know more. She works for the U.S. Geological Survey, or her colleagues like Amanda Demopoulos or Jill.
Yeah, I find it easier to get a geologist than read the paper.
We're also going to keep our eyes out for some brachiopods or shell hash if we come across it as well.
Okay. Is that a ball with yellow things sticking out or just a round rock? Am I seeing things again?
Where's that?
Down to the right.
Yeah. Right under the lasers.
Yeah, that looks like.
This happens to be a rock with some sponge on
Yeah,
it.
small sponges.
Yeah, some small, looks like little sponges. There is a polymastia that grows kind of like that. It will have just little individual oscula that stick up above the sediment.
Hey, Rennie, how far are we from the last transect point?
Not very far.
Not very far, yeah. Okay.
Yeah, we were about 60 meters, 50, 60 meters from it.
Okay. That's what I figured if we were kind of starting back to where we were before.
Yeah, I can put a circle around that so we can see. There's a 100-meter radius.
I wonder why do those sponges close up like that? Just folding in and they can't hold themselves open any longer.
Right.
Cool Argus shot.
Oh, that is great. So for those of you at home, you can choose what channel you're looking at, and you can look at the quad to see the Argus cam, or I believe channel 2 to see just the Argus cam.
There's a really nice, cool little polymap. Yeah, just about to go off the bottom of the screen.
Oh yeah, that sponge that we saw earlier.
Yep. That one looking even more party hat-like.
It's a nice peak here.
We're nose into the breeze. You could drop Argus down a bit if you wanted to.
Roger.
That's how they're streaming behind us.
Yep. I'll come down a few meters.
Yeah.
If you're just joining us, we're diving in the Channel Islands National Marine Sanctuary off the coast of Southern California. And we're at a very nice feature that we've been enjoying for the last couple of hours, around 360 to 370 meters depth. And we have this hard bottom habitat with this nice ledge that almost looks like an extension of a very well-studied reef known as Footprint Reef. That's just south of Santa Cruz and Anacapa Island. Some of our operations today include quantitative transects, which involves moving the ROV in a fixed direction with a fixed height above the seafloor. And then we have analysts that will be watching this afterwards to count and size some of the fish and the corals and the sponges that we're seeing, or at least a subset of them that are bigger than 10 centimeters. We've also done a number of collections. Since we've been seeing so many sponges, we're definitely taking the opportunities to responsibly sample sponges to get a better idea of their taxonomy, since it can be so difficult to identify sponges just from video. We've also fired the Niskin bottles, which collect water samples for a number of different priorities, actually. We're kind of balancing a few objectives, including sampling environmental DNA, which Meredith is in charge of. Taylor Ann, who is the data logger for this wash shift, is also going to be filtering them to quantify potential microplastics that might be found in these areas. And then we're also filtering them to eventually measure particulate organic matter for the U.S. Geological Survey. So we have lots of priorities on this dive. We're also collecting for a number of different partners like the Santa Barbara Natural History Museum. We have a group with the University of Washington that is reconstructing the oceanography and the climatography of this area through stable isotope analysis of some We still have Niskins to fire, is that right, Meredith?
There is one Niskin left. And given that we're seeing a lot of the same stuff we have been seeing, I would actually recommend that that Niskin gets fired kind of mid-water on ascent for the POM group.
Roger that.
Nice little paragorgia, or a nice big paragorgia there. These are bank rockfish that are just kind of hanging in the water column right off the edge there.
I do keep seeing a little bits of the moldy cheese sponge here and there.
Yeah, it's starting to appear more, and I'm wondering if we've just been a little too deep for it in this area, and we'll see more of it as it gets a little shallower.
Mm-hmm.
Yeah, I can't recall what depth we were at when we were seeing it on the express cruises. We also have a number of scientists ashore joining us, so thanks for tuning in.
I think it's a
Nice.
skate.
Skate to the
Oh,
left.
nice.
Another long nose skate.
Skate escape.
Up in the
Skate
water
escape.
column.
Choo.
It saw and heard all the lights and the noise of the vehicle and just went, I'm out.
This is new territory here, is it?
Not quite. We're approaching new territory.
Roger.
Yeah, that trail ran out over there.
We're going up, are we?
Yeah, it seems like it.
Or no?
Yeah, a little bit. It's been pretty steady though.
Maybe the ridge on the left is creating an illusion for me.
Abby says we saw the moldy cheese sponge, and actually the one we collected was at 500 meters, so we are all within the depth area where we thought. So I don't know why we haven't seen much of it until just the last hour or so.
Our oxygen has been steadily increasing since we arrived at this ledge feature.
What's that coral
We're now
in
at
there?
4.2%.
It's I think that's a black coral, and actually, could we zoom on the bright yellow coral or sponge nubs just to the left of it as we come in,
Yeah, okay.
and then we could look at it as well.
Oh, sponge nubs.
Seem to be parked there when the zoom's deep. The little mound just to the left there is what you want to see?
Yeah, the bright yellow little nubs.
Oh, yeah.
Abby had asked if we saw it again to get a zoom on it.
It can zoom in there now. Should get you.
Full zoom.
Roger.
Excellent. Abby says, thank you.
Pleasure.
She said it does look like something different than what we thought it was from the sponge guide we have.
I have to stop him for a minute till I catch up again.
Bridge nav. Hold position.
After seeing so much of these pale yellow Mycale, seeing these just bright fluorescent
Yeah.
sponges, they appear even brighter.
This is new territory.
Awesome.
Bridge, Nav. Uh, 100 meters, 315, speed 0.2. Thank you. Got a skate attack from beneath.
Completely blinded by the lights.
Nice seascape. Onk.
Ship moving, is it?
Yep, just started to back up.
Cover.
I'm also kind of surprised we're not seeing Desmophyllum or any of the other hard cup corals here.
Yeah, there's still time. But you'd think like this is the kind of ledge where typically you'd see Desmophyllum kind of hanging upside down, but there hasn't been any. Maybe the current is just too high and they can't settle there effectively.
Yeah. Just facing the wrong direction.
Mm-hmm. Of course, now we've said that, so you know what will happen is that right at watch change, they'll come around the corner and there will be a bunch.
Yeah, of course. Come down 5 meters, please, Jake.
Yep. Oh.
Good evening, or good morning. Is it morning for you? I think, oh yeah, we got a fresh crew here just chomping at the bit. So I'm gonna find a spot to look at and then hand it over. Thank you everyone for the nice ride. It's been
Thanks, Dan.
fun.
Yeah,
Thanks
thanks for the
Dan.
great flying.
Video's handed over too.
Signing off.
All right, so
I'm
you
gonna
hear some silence as we log off and switch watch leads. So it was a pleasure, everyone. Thank you so much, Meredith. Until next time.
Yep. Thank you. Thank you, Lizzie. And we'll turn it over to Lisa and Jen.
Hey, it's Lisa on. Can you hear me?
Hey, Chad and Lisa, it's Lizzie. Just kidding, I'm back.
Okay, I muted
Great to
everybody
have you,
now.
Chad. I think this is a good opportunity. So I'll just give you a quick rundown. I put a little bit in the chat. Can everyone hear me okay?
Yes, I can hear you
Okay.
fine. Great.
Yeah,
I've
Rennie's
been
been picking
garbled
up
intermittently. Should I be just the Nautilus?
Yeah. Yeah, Rennie says they want to make it to 84, which if we do 0.2 knots is going to be 3.5 hours. So I guess we've been trying to keep the pace going, but I can always hold or adjust the heading. Okay. Yeah, and that's kind of the goal. So, yeah, should be fun. Yeah. I wonder if they found that blue-yellow sponge.
Blue cheese sponge,
The blue
yeah.
cheese, yeah.
Very cool feature. Or 315.
Yeah, we're on 315.
Yeah. Seems like the ridgeline will just keep on 315 for a while.
Yeah. It looks like it bends back a little, but you can see if I need to bring it to like 320 or something, but I think you should have room for a while on this.
All right.
I don't mind. So also it sounds like they did a photo mosaic, and they've been doing, I think, 10 meters by 10 meters. So it's mostly just me sitting pretty and you. So yeah, can try to see what line spacing work for them, but here, actually I can do that.
Are they doing any more transects for this, or are
Yeah,
they?
that's another question. I'm assuming so, but yeah, that photo mosaic is super small. They're doing like 3-meter offsets. So
Roger. Yeah, it looks like we're at the peak.
yeah.
I wonder what this type of crust is.
I know. They look like so much outcrops that were just like mounds of biological stuff on biological stuff.
Yeah.
It's not
cool.
this.
Sorry? Watch the big step. Yeah. Like how it just overhangs like this. It's gorgeous.
This could be a big, cool structure from ocean. Real big.
With a bunch of shadow, though.
Yeah. Get under it.
Do we have our signs ashore? Seems like it's pretty quiet.
Yeah, V-Link is being not its best version of itself.
Roger.
You know, sometimes it works great and sometimes it doesn't. And it just, we cannot figure out why.
Satellites. Yeah. Because we definitely are far enough from the last transit.
Roger that.
It's almost like a parrotfish. What is that? Neat top.
I'm not sure.
No.
Like how there's a starfish just, or a sea star just slumped into that sponge. You had a long day.
Or rockfish.
This is such a cool area. There's so much diversity.
Yeah, definitely. It's a cotton candy thing coming up here. It's kind of cool.
Science on shore is probably like, this is what we've been looking for. Can you hear us?
Marley, you want to take a quick zoom?
Sure thing.
Yeah, this is Chad. I'll be in the back row for most of this watch. The others on shore are having some V-link issues, so they're going to see if it settles out here soon. But I have general guidance for this watch. I know we need to be on
Come wide, please.
deck by 8. And I know we're shallow. What time would pull be?
Sorry?
When do we have to pull?
I think just 30 minutes before. Maybe a little earlier.
Yeah, so we're at 370 meters. We'll probably keep climbing a little bit.
Yeah.
Estimate probably 30 meters to come up, and then another 15 minutes on the surface. So 45, but 45 until, if that makes sense, or 15 after.
7:15. Okay.
Yeah.
Can do.
Also, do you know if they want to keep on doing transects?
Yeah, so basically transects, good targets for the 3D model mosaics that they've been doing this last watch.
Hey, it's Lisa. Can you
Hey,
hear me
Lisa.
now?
Yes.
Yes,
Hey, I had to restart. I'm glad I'm back.
we are too.
Hey, Chad, I'm glad I heard I was on at the beginning when Lizzie was giving you directions or telling you what we still needed. So I heard some of it. Glad. Was Jen able to get on?
She was, and then she was having problems too. So I don't know if she's rebooting now or what.
Okay.
I'm here momentarily, but we'll see how long it stays. So we'll see. But my V-link's been going in and out too. So
Yeah.
we'll do our best here. Luckily, we've got Chad there also, where he's not dependent on this particular piece of technology.
Yeah, we have the real person. So shall we do a round of introductions before we get started, or do you want to talk offline on the separate channel to talk about what we're going to try and do?
Yeah, I'd say, well, I mean, it's your call, Lisa. Whatever you guys want to do. We have transects and I guess mosaics left to do, potential cores as well if we come across some sediment. So if you want to, I mean, you can start a transect and we can do introductions, or we can just keep cruising along here.
Let me just make sure that we understand. So we're on our way to 84. How far are we from that?
84 was just a directional suggestion. We do not need to make it that far.
Bring us to 320.
Okay.
Better.
320 works, yep. 320.
So if is there a 200 meters that we could follow, and everything would be okay for a transect?
Hold on.
I guess I'm asking Nav, Ben, and.
Sorry, I was talking to hold on one second.
Okay.
And this is Jen, and I'm just curious. I think I heard earlier that the circles that are. Oh, go ahead, Ben.
Yeah, so we're in a good position. This ridge is pretty straight, so we're just trying to keep a good speed going to get to the 84 target eventually. But yeah, if you want to do a video transect, we can keep the same heading for way more than 200 meters and still be on this ridge, looks like. We're over 100
So
meters.
Yeah, so I'm watching, so we're just going to be following along this ridge line, running along sort of a contour line, if you will?
Yeah.
Okay,
That's
great.
good. Yeah.
Good.
All right, so
I'd say, let me make sure the transect tracking sheet is open. I'd say we could start one and then we'll do the introductions.
Yeah.
How about that?
Sounds good.
Great, so let's get set up.
yeah, I think 320 might be better. But, yeah. Yeah, that's fine.
So this is the 11th transect?
Yep, that's what I have in high pack. That drive with you, Mary?
Okay.
Yeah, that's what I have.
Okay.
That jives with me.
I see Jen. Yeah, so whenever you want to start, I guess we can start. And then when we're on our way a little bit, we'll do introductions. How about that?
Sounds good. Yeah, I'm going to mark
you.
it.
All right. I'm just gonna escort my camera real quick. All right. And you guys want scaling lasers on?
Yes, please.
Great.
Okay.
All right, I'm gonna start moving forward.
Roger.
All right, science are settling out pretty well. If you want us to go around and say our names.
Sorry, Jessica, were you asking science a question?
Oh no, I was saying we're all settled out now and on our transect, so we can go ahead and do introductions.
Good. Why don't we start with the front row? I think that's how it goes, right? Front seats.
Yeah, I'm Ben Gracian. I'm a navigator mapper.
I'm Jess Sandoval, Sit and Herc, ROV team.
Kim Wallace, ROV team, sitting Argus.
I'm Marley Parker, doing the video engineering.
In the back row, I
And
got myself, Chad King from Monterey Bay National Marine Sanctuary on the vessel.
And I'm Mary Duree, sitting in the data logger seat.
Yay, Mary. I was waiting to hear you pipe up. Oh, hi, and this is Lisa Vonig. I work for the Office of National Marine Sanctuaries at the West Coast Regional Office where I'm the policy coordinator. I have a science background, and it's been really a treat for me to go back to my science roots on these Nautilus dives.
And I am Jennifer Brown. I work with both Monterey and Channel Islands National Marine Sanctuaries with research teams. And I was mostly planning to be engaged in the Central California dives, but given our very flexible situation that we're doing here, I'm happy to get a chance to also help with the science for these Channel Islands dives. So I'll be here with Lisa and excited to be working with all of you. And Lisa and I are both on shore as opposed to everybody else. And we, every now and then, or as we started this watch, I should say, we had some audio issues with our connections to the vessel and the internet, but hopefully that has resolved and we won't have any more of those problems.
Yeah, hopefully.
Chad, or anyone else in the van, can you let us know how our audio is coming through at this point?
We can hear you. There is some static,
Yeah,
sharp noises to it.
I seem to be I'm on the ridge. It's all good.
There's a little bit of finagling I can do to try to resolve that, so I'll keep working on troubleshooting the audio in the background. But the main
Thank
thing is
you.
just keeping you guys connected. So
Yeah.
Thank you. Is it for
Thank
both
you very
of us?
much.
Because I can hear it on Jen's, but I'm not. Obviously, can't hear myself. Is it for both of us?
I can make adjustments individually. So
Okay.
yeah, let me just stand by. Well, just keep talking, communicating. If we need y'all to repeat something, we'll let you know.
Okay.
Okay. So
I'll just try and talk a little more slowly too. I think that helps.
Yeah. So we're on a transect now, our 11th one. We're on what we are calling the Poti Peninsula, and Poti is named after Matthew Poti. He has been working and helping advise us on where to dive. He's collected a lot of data and constructed a habitat model that will help us figure out where are the best places to dive to locate sponges and corals. And on this ridge, he predicted that we would find corals, and his models have been accurate. So that's pretty awesome when these habitat suitability models do work out. And we're pretty excited. I'm hoping he has been able to follow along and see that his work has been fruitful.
Yeah, it's been a really fun dive to watch. I've been having to tear myself away every now and then, but trying to follow as much as possible. Huge sections going through sponge gardens. I see we're back